Good morning, everyone. Today, we're going to talk about digestion and absorption of carbohydrates. I'm Dr. Shreya, Professor and Head, Department of Biochemistry. Now, we'll talk about glucose. Uh, as all of us have discussed already, glucose is the universal fuel for human cells. Glucose concentration in body are maintained within limits. Glucose is the preferred source of energy for most of the body tissues. Brain cells derive the energy mainly from glucose. Fasting blood sugar level is normal individual is 70 to 100 milligrams per deciliter. Okay, this is the thing to be remember that fasting blood sugar level in normal individual is 70 to 100 milligrams per deciliter. When the glucose metabolism is deranged, life-threatening conditions may occur. A minimum amount of glucose is always required for normal functioning. Now, we'll talk about digestion of carbohydrates. The principal sites of carbohydrate digestion are the mouth and small intestine. Dietary carbohydrate consists of polysaccharides, which are examples are starch, glycogen, and cellulose. Disaccharides, sucrose and lactose. Monosaccharides, mainly glucose and fructose. Monosaccharide need no digestion prior to absorption, so they are directly absorbed. Disaccharides and polysaccharides must be hydrolyzed to simple sugars before their absorption. Now, digestion starts in mouth. Digestion of carbohydrates begin in the mouth. Salivary gland secrete alpha amylase, which initiate the hydrolysis of alpha 1,4 glycosidic bonds of a starch and glycogen to dextrine. Now, digestion in stomach. Carbohydrate digestion halts temporarily in the stomach because the high acidity inactivates the salivary alpha amylase. Digestion in intestine. When acidic stomach contents chyme reach the small intestine, they stimulate mucosal cell of duodenum to release secretin and cholecystokinin (CCK) that stimulate pancreas to release pancreatic juice into intestinal lumen. Second one, secretin stimulate the release of bicarbonate which neutralize the acidic chyme. Third one, that is CCK cholecystokinin. It stimulates the release of digestive enzymes. Now, pancreatic alpha amylase degrade dextrine further into maltose and isomaltose and limit dextrine. The cells of brush border of intestine contain the enzymes sucrase, maltase, isomaltase, and lactase. Now, you can see the reaction that the maltose is acted upon by maltase to form glucose and glucose because it is a disaccharide composed of 2 units of glucose. So, it is broken down to glucose plus glucose. And second one, isomaltose, it is again at the branching point. So, it is broken down by isomaltase to glucose plus glucose. The sucrose is acted upon by sucrase to which is broken down to glucose plus fructose. And lactose is acted upon by lactase to form glucose and galactose. And alpha limit dextrine, it is acted upon by dextrinase. So it is converted to glucose plus maltotriose. All the above enzymes except dextrinase hydrolyze alpha 1,6 bonds. End products of carbohydrate digestion are glucose, fructose, and galactose, which are readily absorbed through intestinal mucosal cells into bloodstream. Now you can see this is the chart where the dietary carbohydrates, they are polysaccharide, disaccharides, and monosaccharides. Polysaccharide, which is starch, it is acted upon the mouth by salivary amylase to form dextrine. In stomach, there is no digestion, and dextrine comes directly into the small intestine, where it is acted upon by pancreatic amylase, and which is converted into glucose. Okay, first maltose and then maltose, and by intestinal enzyme maltase is acted upon um, maltase acts on maltose to convert the maltose into glucose units. Okay, and disaccharides, in case of disaccharides, these disaccharides like these disaccharides, they are like maltose, sucrose, and lactose. They are acted upon by maltase, sucrase, and lactase to convert into glucose, fructose, and galactose. Similarly, monosaccharides, they are present in the diet. They are glucose, fructose, galactose. They are directly absorbed. Okay, so uh, this is about the picturization or chart form of dietary carbohydrate and their digestion. So also again a picturization of digestion, like various mouth, stomach, pancreas. You can draw this, describe the diagrammatic representation of digestion of carbohydrates, polysaccharide, disaccharides, and monosaccharide units. Now come to the absorption of carbohydrates. Carbohydrates are absorbed as monosaccharides from the intestinal lumen through the mucosal epithelial cells into bloodstream of portal venous system. Now, mechanism of absorption of monosaccharides. There are two mechanisms, like first one, that is active transport, second one facilitative transport. Active transport with concentration gradient and facilitative transport with concentration. Now we will see the difference. Active transport. The active transport of glucose and galactose across the brush border of mucosal cells occur by active transport. It is an energy requiring process that requires transport protein and presence of sodium ions. A sodium dependent glucose transporter, that is SGLT1, binds both glucose and sodium at separate sites and transports them into the cell. The sodium transport down concentration gradient and glucose transported against concentration gradient. This process is called as co-transport or symport mechanism. The energy for this reaction is provided by ATP linked to sodium pump. Okay. This is the picturization of absorption of carbohydrates. You can see, you can draw the picture, sodium, potassium ions. This is about the absorption of carbohydrates. Okay, you can see it, you can draw it by writing the answer for describe the digestion and absorption of carbohydrates. Okay. This is again the picturization. Okay, root mechanism. Transport by root mechanism. This is the increased glucose concentration gradient. Okay. Glucose transporter. Root mechanism, they are the glucose transporter. Group 1, group 2, group 3, group 4, group 5. Okay, you can just see facilitative biodirectional transporters. Group 1, they occur in brain, kidney, colon, placenta, and erythrocytes. What is the function of group 1? Uptake of glucose. Then group 2, they are present in liver, pancreatic beta cells, small intestine, kidney, rapid uptake and release of glucose. And group 3, present in brain, kidney, placenta. Their function is uptake of glucose, and group 4 present in heart and skeletal muscle, adipose tissues. Their function is insulin stimulated uptake of glucose. Then this is now there are various disorders of digestion and absorption of carbohydrates, bacterial fermentation in large intestine, the production of H2 and CO2 gases, low molecular weight acids like acetic acid, propionic acid, and butyric acid, which are osmotically active. Then abdominal cramps and osmotically active products draw water from the intestinal cell into human, resulting diarrhea and dehydration. Lactose intolerance, deficiency of enzyme lactase, sucrase deficiency, inherited deficiency of the enzyme sucrase. Okay, now carbohydrate digestion and absorption is so efficient in healthy individuals. All dietary carbohydrates absorb, the lower jejunum, predominantly monosaccharides are absorbed. Okay, you can see the picturization, efficiency, lactase, lactose intolerance. Okay. It is a kind of soda, lactose intolerance.